The overall goal of the following experiment is to test for specific variants in the BRCA1 protein using an in vitro homologous recombination assay. This is achieved by replacing the endogenous wild-type BRCA1 protein with the variant being tested through the transfection of both short interfering RNA specific to BRCA1 and plasmid for re-expressing test BRCA1 variant proteins into cells. As a second step, the short interfering RNA and the plasmid for expressing mutant BRCA1 is retransfected in addition to a plasmid for expressing the ISCE1 protein, which causes DNA damage at a specific site in the DNA genome of the test cell. Next, the cells are incubated for 2 to 3 days in order to allow for the DNA damage and repair to occur. Results are obtained that show whether specific protein variants of the BRCA1 protein function in DNA repair by homologous recombination based on flow cytometry analysis of GFP-expressing cells.
This technique combines two established methods in order to create a versatile system for analyzing specific protein variants in the repair of DNA damage by homologous recombination. The main advantage of this technique over existing methods is that previously established methods relied on cell lines that had a genetic knockout of BRCA1. We use a more manageable cell line derived from HeLa cells that enables BRCA1 depletion by RNA interference of the endogenous protein and add back of test protein. In these cells now expressing a variant BRCA1 protein, we can test for homologous recombination.
Generally, individuals new to this method will struggle because of excessive cell death during the procedure. It is important to have a transfection condition carefully set, maximizing the effective recombination reaction while limiting cytotoxicity.